This lab is located in the biochemistry building at Purdue University. In our laboratory, we actually are a hybrid molecular biology, biochemistry, molecular genetics lab. We sort of have this large toolbox, and we select the right tool for the job. What it means to be a geneticist is that you work with a model organism that's living, and you manipulate the genes within that organism in order to study complex problems. This is our fly incubator. This is where we raise the majority of our fly stocks. We have them growing at 25 degrees, and we have them kept in racks like this where we have 40 vials per rack on the average. In the type of genetic research that we do, we can make mutations in an animal model system and then study how those mutations affect the life of the animal, development of the animal. These flies have a point mutation which renders them temperature sensitive, and simply using my body heat, if I just warm them up, and as a result the animals become paralyzed. Most people, when they first start working with flies, or before they're exposed to it, have this opinion that flies are dirty. If you look at a fruit fly under a microscope, you'll see that they are actually very clean little animals. They're constantly cleaning themselves.
So I'm putting CO2 into the vial to get them to go to sleep so I can move them around a lot better. So I put them on the fly pad, and so as long as they're on there, they will stay asleep for me.
The reason why we study fruit flies is because fruit flies have brains, per se. They actually have neurons that wire together to form a brain, just like you and I. We're trying to understand how the brain forms connections. The brain is composed of a number of small cells called neurons. Neurons are elongated structures, much like extension cords. In order for the brain to work properly, all these connections have to form in a very, very precise manner. What we've found is actually quite astounding. We found that these molecules that function to help neurons recognize one another have a function later in life which appears to keep these neurons alive. And we find that many of these genes that function within this particular pathway that we're studying are genes that are involved in neurodegenerative diseases in human beings, including Alzheimer's disease, Parkinson's disease, and Lou Gehrig's disease. This research actually has a good deal of significance for me because my father passed away having Parkinson's disease, and I saw him slowly, transition from a father that I knew and could interact with and do stuff with, and go out with, and to somebody that ended up being bedridden. So it's very important to me. It's very significant, to me, to be able to address this question. It's the ability to see something that affects you deeply, and then have the opportunity to be part of the solution. When we make a discovery like this, it's actually first of all, it's a very exciting time in the lab. Okay? It's one of the most exciting times I've ever had in my career. To make a big connection like this is amazing. To actually think that my Drosophila research can have a broader impact on human health by studying the genes that are required to keep these neurons alive in these neurodegenerative diseases. Right now, what we're interested in doing is further characterizing how these molecules function to keep neurons alive with sort of our eye towards the future. How could we design drugs to alleviate suffering for those that have Alzheimer's, Parkinson's, or Lou Gehrig's disease? And I think it's a serendipity in science that you have to be open to. You have to realize that you're not going to always get what you want, but sometimes what you do get is 10 times better than what you wanted. I got into science at a very early age. When I was a Boy Scout, we did some merit badges in science. Obviously through school, I was very much interested in the science classes. After Boy Scouts, I was a medical explorer scout, and then I came to college and immediately got involved in the science program here at Purdue University, as a matter of fact. The idea is to become a scientist, and in order to become a scientist, you need to formulate a hypothesis. You say, I think that this is the way something is, okay? and then you design experiments that test that hypothesis. And you have to realize that many of your hypotheses are going to be wrong, okay? Don't get all distraught by coming up or finding out that your hypothesis was not correct, okay? That's a big part of science. A big part of science is being wrong and learning that you're wrong, that's not the way you thought it was, but it's a different way, okay? And that's what sparks creativity in an individual, okay? And once you have that creativity and you think about these problems, you immerse yourselves in these problems, you end up coming up with these big aha. That's what it is. And it might not even be what you were looking for in the first place.